It is just past 5 in the morning, and the garden is still. The light has not yet decided what it wants to be, gray at the edges, thin and colorless above the rooftop. A single blackcap warbler sits in the elder tree at the back of the yard. You have seen it there before. It arrives in spring, stays the summer, and then one day in autumn it is simply gone. No ceremony. No pattern you can see from the kitchen window. It weighs less than a 20-pence coin. It has traveled, at some point in the last 6 months, more than 3,000 miles across open water, desert, and mountain range. It did this at night. It navigated without instruments. It returned, not to the same country, not to the same county, but to this garden, this specific elder tree, this exact branch. This morning, watching it preen in the pale light, you probably feel nothing more than mild pleasure. But there is something happening inside that bird's skull that no laboratory on Earth has been able to reproduce. That is not a poetic exaggeration. It is the precise state of the science. Migratory birds perform feats of navigation, computation, and biological endurance that researchers have documented in detail across decades and hundreds of peer-reviewed studies. The instruments exist. The data exists. The mechanism, the actual physical, chemical, cellular process by which a bird weighing 11 grams finds a specific hedgerow in the south of England after a non-stop flight over the Sahara, remains, at its core, unsolved. These are not mysteries of observation. They are mysteries of explanation. Scientists can watch what birds do. They cannot yet fully explain how. This channel is dedicated to exactly this category of scientific strangeness, things that are confirmed, measured, and documented, with answers that remain incomplete. If that kind of question is one you find genuinely interesting, subscribe. The final section of this video contains one particular fact about a bird's capacity for time measurement so specific and so strange that I am still not sure what to do with it. I will not reveal it yet. Stay with me. Most people who watch migratory birds assume they navigate the way we do, by learning landmarks. They see the same mountains on the way south that they saw on the way north. They remember rivers, coastlines, the shape of a bay. This is a reasonable assumption. It fits with what we know about animal memory and spatial learning. It is also substantially wrong. In a now famous series of experiments conducted at the Max Planck Institute for Ornithology in Germany, researchers fitted European robins with small magnetic coils that could alter the local magnetic field around the bird's head. The robins, placed in orientation cages, small funnel-shaped containers where a bird leaves scratch marks indicating which direction it wants to travel, immediately reoriented when the artificial field shifted. They were not navigating by landmarks. They had no landmarks. They were responding to something they could sense in the field of the Earth itself. The robin possesses magnetoreception, the ability to detect the inclination angle of the geomagnetic field, the ratio of the vertical to horizontal component of that field, and use it to determine approximate latitude. Researchers at the University of Oldenburg have identified cryptochrome proteins in the robin's eye, specifically in a region called cluster N of the forebrain, that appear to interact with the geomagnetic field via a quantum mechanical process involving electron spin. A bird appears to literally see the Earth's magnetic field overlaid on its visual field, like a compass drawn in light. What makes this genuinely strange is this: no one has successfully replicated a quantum-coherent biological compass in a laboratory. The leading model, radical pair chemistry, in which pairs of electrons maintain entangled spin states long enough to register a directional signal, requires quantum coherence to persist in a warm, wet, chemically noisy biological environment for at least a microsecond. Most physicists had assumed this was impossible. In birds, something appears to make it work. The exact protein structure, the precise mechanism of signal transduction from the eye to the brain, the way the information is interpreted and acted upon, none of this has been reproduced outside a living organism. The assumption most people carry about bird migration is that it is seasonal. Birds go south when it gets cold, north when it warms. Temperature is the trigger. This is intuitive and it is broadly directionally correct, and it is also far too simple. In 2007, a research team at the University of Lund in Sweden conducted a long-term study on blackcaps, the same species perching in your elder tree this morning. They found that blackcap warblers possess an internal calendar so precise that individual birds begin physiological preparation for migration, laying down fat reserves, altering sleep patterns, producing elevated melatonin, within a predictable window of 4 to 7 days, year after year, regardless of temperature variation, food availability, or light levels within normal seasonal ranges. This internal calendar is not simply a response to day length, though photoperiod plays a role in calibrating it. It is endogenous, generated from within. The birds used in the Lund study were kept under constant artificial lighting conditions. Their migratory restlessness, measured by activity monitors and called Zugenruhe in the technical literature, still began on schedule. What makes this genuinely strange is this: the molecular machinery of this internal annual clock, how it resets each year, how it retains its precision across a lifespan, how it maintains accuracy through temperature fluctuations that would disrupt most chemical processes, is not understood. We know the clock exists. We have measured its outputs with considerable precision. We do not know how it keeps time. There is a common belief among birdwatchers that birds returning to breed in familiar territories are using memory, that they know the way home because they have been home before. This is true, and it is also only part of the story. In 2015, researchers at the Max Planck Institute tracked first-year bar-tailed godwits, shorebirds making their inaugural migration with no prior experience and no guidance from adult birds, on their first southward journey from Alaska. These birds had never made the trip. They had no learned route. They flew non-stop across the Pacific Ocean, covering up to 11,000 kilometers in a single flight, and arrived within a small target zone along the coast of New Zealand. First-year birds. Alone. No map. No experience. Correct destination. This means that some component of the navigation is genetically encoded, not learned but inherited. Researchers believe the initial migration vector, a combination of compass bearing and approximate distance, is transmitted through genetics. But the precision of the arrival cannot be explained by a simple inherited vector alone. The wind patterns over the Pacific shift. Storms push birds off course. Yet first-year godwits correct for these deviations and arrive within the target zone. What makes this genuinely strange is this: the mechanism by which a naive bird recognizes that it has drifted off a course it has never previously flown, and the mechanism by which it calculates the correction required to return to a target it has never seen, has not been identified. The navigation system appears to contain something that functions like a map. But where that map is encoded in the bird's brain or genome, and how it is read, remains open. It is commonly understood that birds burn fat to fuel long-distance migration. This is correct. What is less commonly known is the biological scale of what that actually involves. A ruby-throated hummingbird weighs approximately 3 grams. Before its autumn crossing of the Gulf of Mexico, a non-stop overwater flight of roughly 900 kilometers, it doubles its body weight in fat. Those fat reserves, approximately 1.5 grams of stored energy, will power 11 hours of continuous flight across open water with no opportunity to land, feed, or drink. That is already remarkable. But consider what happens at the cellular level during this process. During preparation for migration, birds undergo a process called hyperphagia, a period of accelerated food intake that can increase caloric consumption by a factor of 10 compared to normal. The liver doubles or triples in size to facilitate fat processing. The pectoral muscles, the primary flight muscles, increase in mass. And simultaneously, organs not needed during flight, the digestive tract, the kidneys, the reproductive organs, shrink. A migratory thrush can reduce its gut mass by 40% in 24 hours to save weight. Pause on that number. 40%. In 24 hours. Then, on arrival, those organs regenerate. A bird lands after a multi-day flight and begins rebuilding its gut in real time to process food. This capacity for rapid, reversible organ atrophy and regeneration is called phenotypic flexibility, and it is unlike anything observed in mammalian physiology. Humans who experience significant muscle loss require weeks or months of rehabilitation. A garden warbler does it on a schedule, repeatedly, year after year. What makes this genuinely strange is this: the molecular signals that initiate and terminate organ regression and regeneration in migratory birds, the precise hormonal and genetic cascade that tells the gut to shrink by 40% and then rebuild it intact, have not been mapped in sufficient detail to replicate in any other system. The same signals that tell a blackcap's digestive tract to disappear and then regrow are not understood well enough to be reproduced outside the bird. There is an assumption, a natural one, that when scientists speak of bird navigation, they mean a single system. One compass. One mechanism. This is perhaps the most significant misconception in the field. Birds do not navigate with one system. They navigate with at least four, simultaneously, and integrate conflicting information from all of them in real time. The geomagnetic compass, described in Section 1, reads the inclination angle of the Earth's field. A second magnetic sense, believed to involve iron-containing crystals in the bill, may read field intensity. The sun compass, calibrated by an internal clock, uses the position of the sun and the time of day to derive direction. The star compass, used primarily on clear nights, reads the rotation point of the night sky rather than individual constellations, allowing it to remain accurate as the season and the visible stars change. Additionally, birds appear to use infrasound, acoustic waves in the frequency range of 1 to 10 Hz generated by ocean waves, mountain ranges, and atmospheric events, as a positional cue detectable at distances of thousands of kilometers. Researchers at the University of Auckland published work in 2022 showing that bar-tailed godwits adjust their navigational weighting between magnetic and celestial cues depending on conditions, prioritizing star-based information on clear nights, shifting toward magnetic sensing under cloud cover. The integration is dynamic and context-dependent. What makes this genuinely strange is this: no model has been produced that successfully predicts real bird flight paths by integrating these 4 or 5 systems as the bird appears to integrate them. Each system is partially understood in isolation. The algorithm, if algorithm is even the right word, by which conflicting or ambiguous signals from different sensory modalities are resolved into a single navigational decision, remains uncharacterized. We do not know what happens when the magnetic field says north and the stars say slightly west of north and the infrasound says something else entirely. But the bird knows. The final thing. The one I mentioned at the beginning. Most observers assume that a migratory bird's knowledge of when to leave and when to arrive is a general, seasonal sense, a rough feeling for time of year. What the research shows is considerably more precise than that. A research program at the University of Groningen in the Netherlands followed individual pied flycatchers across multiple years at a breeding site in the Netherlands. Pied flycatchers are long-distance migrants, wintering in West Africa and returning to breed in Dutch woodlands. The timing of their return is critical. They must arrive to coincide with the peak emergence of winter moth caterpillars, which represents the optimal feeding window for chick growth. This peak is temperature-dependent and shifts year to year. The birds arriving earliest at the breeding site were not necessarily the best performers. The birds that bred most successfully were those whose arrival date most closely matched the caterpillar peak in that specific year, a peak they could not have known in advance, since they were in sub-Saharan Africa when it occurred. Some individual birds, tracked across multiple years, showed arrival dates that tracked the caterpillar peak with a precision that cannot be explained by the bird responding to local conditions on arrival. They would have needed to begin their journey weeks earlier, in Africa, at the right time to arrive in the Netherlands on the right day for a food emergence event they had not yet witnessed. One individual female was tracked for 7 consecutive breeding seasons. In 5 of those 7 years, she arrived within 3 days of the local caterpillar peak. She was doing this from 3,000 miles away. What makes this genuinely strange is this: the mechanism by which a bird in sub-Saharan Africa appears to time its departure to match a temperature-dependent insect emergence in a Dutch woodland, an event that varies by weeks between years, is not understood. There is no identified sensory cue, no known proxy variable, no satisfactory model. The correlation exists. The data is robust across hundreds of individual birds and multiple decades. The explanation has not been found. It is 5 past 5 now, and the light in the garden has shifted. The blackcap is still in the elder tree, working through its morning preening with the methodical attention of something that has important business later. In a few weeks, it will be gone. It will fly southeast over the Channel, cross the Mediterranean, cross the Sahara, and reach the savanna of sub-Saharan Africa, a journey of 3,000 to 4,000 kilometers completed largely at night, using a quantum compass in its eye and a clock whose mechanism no one has yet described. In the spring, if all goes well, it will return. Possibly to this garden. Possibly to this tree. You cannot see any of this from the kitchen window. But you know it now. And knowing it changes what you are looking at. The garden has not changed. The bird has not changed. But there is now a sense, watching it, of standing at the edge of something, a set of capabilities so refined and so poorly understood that the most sophisticated navigation technology humans have built cannot replicate what is sitting in an elder tree, preening. The bird finishes, rouses its feathers once, and is still. Whatever it knows, it is not telling. Have you ever noticed a migratory bird return to a specific spot in your yard, the same tree, the same fence post, the same corner of the garden, year after year? How many years in a row, and do you know what species it was? I read every one.